Hello everyone. In this video we are going to discuss fungi, and we are going to start with the general characteristics of fungi. So let's start. So let's remind ourselves that fungi are part of the eukarya, so they are eukaryotic organisms. In terms of the pronunciation, you may find differences in the way that people pronounce the name. Some people pronounce it fungi, some others pronounce it fungi. You may even find people pronounce it fungi. I'm more comfortable in pronouncing it as a fungi. That's why I'm going to use this pronunciation throughout the video. So fungi are parts of the eukaryotic organisms, and the study of fungi is referred to as mycology. So whenever you see this word in textbook, it refers to the branch of science that focuses on studying the fungi. So let's move on to the general characteristics of fungi. They are chemoheterotrophic organisms. They are chemoheterotrophic eukaryotic organisms, meaning that they don't produce their own food. They get their food ready, and usually they are decomposers, if you remember when we discussed that with bacteria. So bacteria and fungi are the main decomposers, and they have a chitin cell wall. Also, that was discussed earlier, that the cell wall of fungi is made of chitin in addition to some other glucans or sugar molecules. So this is something characteristic for fungi, the chitin cell wall, and they are organisms that produces spore, but spore formation in fungi is produced for the aim of reproduction, and that's one difference between the spores of fungi and the endospores in bacteria. So if you remember, endospores in bacteria are not for reproduction purposes. They are for survival purposes. They are resistant structures made just to bypass a period of unfavorable conditions. Then once the conditions are suitable again, the bacteria will start to grow again and form the vegetative cells and so on. So it is not a reproductive structure in bacteria. However, in fungi, the spores are made for reproduction. Some other differences between bacteria and fungi is the pH that they prefer for their growth. So usually bacteria are mostly neutrophils, meaning that bacteria prefer like a neutral pH around pH 7. So let's say something around 6 to 8, this range. Most bacteria will grow preferably at this kind of pH. Fungi, they prefer the slightly acidic pH, so pH 5 or pH 6. This is the preferred pH for the growth of fungi. They can grow in low moisture. They can withstand high osmotic pressure environments. And this is actually one of the applications of having the chitin cell wall, one of the impacts of having a chitin cell wall, that it supports them and give them resistance against high osmotic pressure. And because of that, fungi can grow on like jellies, they can grow on salted food. They actually have a very high tendency to grow in most different types of nutrients, and we will see this in the next slide as well. As I said, they have a very high tendency to grow on diverse types of nutrients, and this is also facilitated by having the ability to metabolize even the different or the difficult types of carbohydrates, the complex carbohydrates. So, and again, this is one difference between fungi and bacteria. Bacteria usually prefer the simple sugars like glucose, fructose, sucrose, and so on. But when it comes to fungi, even the complex carbohydrates like lignin, for example, they are able to digest this complex carbohydrates, and that's why they are able to grow on them, and that give them an advantage when it comes to growing on different media. Fungi sometimes usually take longer time to grow in the media, and if you think about all of these, you may find why in the labs, for example, in medical labs or clinical labs, the physician should advise the technician working there, if the physician is suspecting a clinical sample containing fungi and he is interested in knowing if there is a fungi there, so he should advise the technician there to grow the specimen on media that supports the fungi. Why? Because, as we said, they prefer an acidic environment. They can grow on complex carbohydrates media. So the requirements for growing fungi can sometimes be different than the requirements to grow a bacteria, and for that reason, the media supporting the growth of fungi is not always the same media that supports the growth of bacteria. Yes, fungi can grow on the same plate, but if you want to grow them specifically, and the optimum conditions for fungi are a little bit different than bacteria. That's why it's better for the physician to mention that he's interested in examining whether a fungi is there in the specimen or not, because that may mean that the technician may use another more specific media for supporting the growth of fungi. Okay, so these are the general characteristics, and now we are moving on to the vegetative structures and the characteristics of these vegetative structures of fungi. So some fungi exist in the form of yeast, and yeasts are unicellular, non-filamentous fungal structure. They multiply by budding, and other fungi, they exist as what we call molds. So molds are the multicellular, the filamentous type of fungi, and some fungi even exist as yeast and as molds, and they can interchange their state from yeast to mold depending on the temperature of the growth. So these fungi, which are able to exist as both forms, we call them dimorphic fungi. And as you know, di means two, morphic meaning like morphological state. So these fungi exist as two morphological states, the yeast state and the mold state, and the difference is depending on the growth temperature. If you grow them at 37 degrees, they will exist at the yeast form. If you grow them at 25 degrees, they will exist as the mold form. So this figure highlights this fact and highlights that they can interchange their state. So it's like a reversible characteristic. They can exist as the yeast form at 37 degrees. So if you isolate these fungi from in vivo specimen, if you isolate them from a living organism, for example, they will exist in the yeast form, because the temperature of the living organism will, if you are talking about human, it will be at 37 degrees. And if you isolate them and grow them in the lab at 25 degrees, this state will be the common one, which is the mold form. So back to the mold form, we said that the mold form is the multicellular, the filamentous type of fungi. The body, so here we are going to highlight some terminology and keywords. So the body that we see is known as the mycelium, and the plural of that is mycelia. So whenever you see a fungal growth on, for example, a plate like that, so this is the mycelium, and this mycelium is made up of individual strands. Each one of these strands is called hypha. So if you are talking about more than one of these strands, the plural form is the hyphae. So think about it as a carpet, for example. So the carpet, the whole structure is the mycelium, and individual threads that makes this carpet is the hypha. So if you are talking about 2 or 3 or multiple threads, you are talking about hyphae. So these are the individual threads, these are the individual strands making the fungal body, which is the mycelium. So mycelium is a collective mass of hyphae, and hyphae are the filaments. These hyphae can be septated, meaning that they are divided by cell walls, or they can be non-septated, so non-divided. And you can see it here in this picture, and in the next slides we are going to see more clearer images. So here we see septum. So we have many septums separating the individual cells, so the cytoplasm is not continuous and the cells are separated with these cross walls. So we call this a septated hyphae. Some other hyphaes, they don't have these cross walls, they don't have these septums, and because of that the cytoplasm is continuous, and we call them, they are cenocytic, or they are non-septated. So this is a division of hyphae based on the presence or absence of the cross walls septa. Another type of division, you can classify this hyphae into one of two types. The first one is called vegetative hyphae. So these work in a similar way to the roots of a plant. So their role is to go deep to absorb the nutrients. And the other type is called the aerial hyphae, and as the name implies, this is a type of hyphae that will grow in the air with the function of dispersing the spores, which could be sexually spores or asexually produced spores. So if we can represent that, so imagine this is fungi growing on a surface. It could be a surface of growth media in petri dish like that, or could be a surface of a media, for example, in a piece of bread. So the collective mass of all these hyphae, we call it a mycelium. So the growth that we see on the piece of bread is a mycelium. This mycelium is built up of hyphae. Some of these hyphae go down, they penetrate the surface of the media to absorb. So they go under the surface of the media to absorb the nutrients. We call them the vegetative hyphae. Others, they grow up on the surface of the media in order to disperse the spores. We call them the aerial hyphae. Again, highlighting the fact that fungal spores differ from bacterial spores. As we said, the fungal spores are reproductive structures. They are carried by the aerial hyphae in order to be dispersed. And actually we have a diverse spore forming structures, and they have different morphologies, and fungi can be classified based on their spores and their spore forming structures.